Professor Yao, distinguished professors, distinguished guests, students. It's my honor and pleasure to talk a little bit about my work today. So I just want to show my slides. So this is a picture of myself actually over 30 years ago when I was still a medical student at Oxford. So as a medical student, we have to study different subjects. And one of the subjects was obstetrics. When I learned about how when a woman is pregnant, she would like to know about the genetic health of the baby. But at that time, the most accurate method is to introduce a needle into your uterus and to take some fluids, a process called amniocentesis. But every time we do that, there's a risk that we might harm or even kill the baby. So as a young medical student, I was thinking, is it possible to develop a non-invasive blood test to do away with all that risk? So I was thinking, maybe it's possible that the baby would release its DNA in the bloodstream of mother. But the question is, how do you prove that? So I was thinking, well, if the baby is a boy, maybe we can start to see male chromosome DNA in the blood of the mother. But at that time, I have no idea how do I actually go and extract this DNA from the plasma. But I remember that being in college, of course, the college food is not entirely good. So sometimes I'll actually go back home to cook instant noodles. But of course, you can imagine when I cook instant noodles, basically you boil some water and then you put the noodles in. And then a few minutes later, you have your dinner. So I was thinking, well, is it possible I can just boil some of the blood plasma for the mother and see what happens? But surprisingly, even with this very crude preparation, when I put a few drops of this boiled soup into a polymerase chain reaction to amplify Y chromosome DNA, I start to see signals in the blood of some women, as indicated by this white band here, and no signals in others. And eventually I find out all the women with signals all had baby boys. So this is the first demonstration of the presence of fetal DNA in maternal blood. And it was very counterintuitive because we all thought that DNA should be inside cells rather than floating outside cells. And then we realized that actually this fetal DNA is present very early, like the 7 weeks of pregnancy. And actually by the 10 weeks of pregnancy, the concentrations reach some 15%, which is amazing because imagine the baby so small and now mother so huge. And furthermore, after delivery of baby, the DNA is gone within 2 hours. It's almost like the baby is sending a message to the mother, but the mother is clearing that message as soon as pregnancy finishes. And then we thought, well, we can use this new source of fetal DNA to do some prenatal testing. And we start with something simple. So we thought, well, can we use that to determine the sex of the baby, which is important because certain genetic disorders predominantly affect boys, like hemophilia, like certain form of muscular dystrophy. And then we got that done, nearly 100% accurate. And then we try to see if we can test the fetal blood group type, because sometimes if the mother and fetal blood group type doesn't match, the mother could produce antibody to attack the baby. And this, once again, we achieved with close to 100% accuracy. And then we decided to be more ambitious. Now, the number one reason why pregnant women go for prenatal testing is because they are worried the baby might be suffering from Down syndrome. So Down syndrome is caused by a baby having an extra copy of chromosome 21, as indicated here. So usually we have 2, but in a Down syndrome baby, it has 3. And usually you typically analyze somebody where someone has Down syndrome by taking a cell from an individual, and then you're counting how many chromosome 21 is within that cell. But the problem in my field is that I don't have cells because the DNA are swimming outside cells. So how can you do that? So it took us 10 years to actually work this out. So eventually the solution is something like this. So let's say that you have a wallet that you put in different number of coins in different days of the week. If I want to know how many coins you have in your wallet, the simplest way is to ask you to hand me your wallet. And that would be like invasive prenatal diagnosis. But there's a non-invasive way. And that is if I ask you to go and stand on a balance. Now, theoretically, on the day when you have more coins in your wallet, you should be a little bit heavier, assuming that your balance is precise enough. And so essentially here, we have to build a molecular balance to see the chromosome dosage in the babies. So basically what we do is that we sequence millions of DNA molecules in the blood so as to count them. And because I know the human genome sequence, I can map each of the sequence to the chromosome from which it comes from. And then I can tally very accurately the relative number of those chromosome fragments. And interestingly, when I did that, I find that the result to be amazingly accurate. Now, for example, here you can see that the green and the red signals are from baby with Down syndrome. We also call that trisomy 21 because there are 3 copies of chromosomes 21. But on the other hand, if the baby is normal, as indicated by the blue and this orange signal, then it is actually very low level. And then being very excited, then we decided to do a clinical trial to prove that this would work on a large scale. And we finished the trial in 2011, and the accuracy was really amazing. It's over 99% accurate because it's so accurate that actually within 10 months after we published that trial, the test was launched clinically. Now, to date, the test is called NIPT, or non-invasive prenatal testing. It's now done all over the world and used by millions of pregnant women every year. Now, it seems that the goal that I set for myself when I was still a medical student has largely been done. So the question is, what else can we do? Now, if we are being very ambitious and push this to the limit, then of course we wonder, well, can I use this technology to sequence the entire genome of the baby? And this is a tough nut to crack because the human genome actually consists of some 6 billion base pair. And also because this genome is fragmented into millions of pieces and also is mixed in with the mother's genome in the mother's blood. So initially I have no idea actually how do you do this until actually one day I went to see this Harry Potter movie with my wife. And this movie is quite interesting because the first 10 minutes of it was in 3D. So I remember I was sitting in a movie theater, put on my 3D glasses and waiting for the movie to start. And then suddenly I saw the Harry Potter title flying towards me. And then my eyes was actually drawn to this H here in 3D, which surprisingly in 3D actually looks to me like a pair of chromosomes. And then suddenly I have an inspiration. I thought, well, that for every pair of chromosomes the baby has got, it has 1 copy from father and 1 copy from mother. So maybe I can crack the fetal genome with 2 algorithms, one for the father side and the other for the mother side. So let's say this picture is the father's genome, and this one is mother's genome. So baby will have 1 half from father and the other half from mother. So now remember that 2 algorithms. So the first one to crack the father's half of the genome. So imagine basically that in the mother's blood, there's a lot of mother's genome floating around, and then we're trying to crack the father's half of fetal genome. So how do you do that? So basically you compare the father's genome and the mother's genome, and then try to find things which is only present in the father's side, but absent in the mother's side, but which is easy to find, like the flower. And then we go into the mother's blood to hunt for the flower. So every time you find that flower, that is a little bit of the stuff that the baby has inherited from the father. And then you zip all those flowers together. So out come the father's half of fetal genome. Now, how about the mother side? And the mother side is more difficult because the baby is swimming in this ocean of mother's DNA. So anything that the baby has that is coming from the mother have it herself. So you cannot differentiate the two. So you have to do something smarter. So what I was thinking is that the mother, if you look at her genome, the left-hand half and the right-hand half should be present at a ratio of 1:1, just like my left hand and my right hand. So now imagine if the mother passes the right-hand half to the baby, and the baby releases it back in the mother's blood. So inside the mother's blood, there should be more right-hand half compared with left-hand half. So basically what you do is that you go into the mother's blood, and then you count the number of halves. And the half which is in slight excess is a half inherited by baby. And then you do it millions of times. So the question is, does this work? So this work, we recruit a couple from the Prince of Wales Hospital in Hong Kong. The mother is 12 weeks pregnant, and both the father and mother are carrier of a genetic form of anemia called beta thalassemia, which is very common in this part of the world. And then we take her blood and then do some sequencing. So just now, when I told you, when we did the Down syndrome test, you do maybe a few million molecules and your test is 99% accurate. But now with this project, we have to sequence billions of DNA molecules. And actually this case cost me about $200,000 to do this one case. But it's important because we're trying to demonstrate something in principle for the first time. Now, so this is so-called circles plot in which the chromosome is plotted in a circular format. And then this blue signal is the fetus genome. So you can see basically entire fetal genome is in the blood. And then we can zoom into any region of the genome we want. So in this case, the gene for beta thalassemia is on chromosome 11. We zoom in, and lo and behold, we find the baby is a carrier of beta thalassemia. So now the baby is healthy. So this work was published and was shown on the cover of this magazine. So then we thought, well, this fetal technology seems to be doing well. So what other branches of medicine can we apply it to? And I was thinking, well, the baby growing inside mother is a little bit similar to a cancer growing in a patient. So maybe my method can be used for cancer as well. So we decided to take a look at this cancer called nasopharyngeal cancer, or NPC for short. This is a cancer which is affect the head and neck region over here, which is particularly common in the southern part of China. And one reason why we choose this cancer is because for this approach to work, I need to have a DNA species which is specifically associated with cancer, just like the Y chromosome is specifically associated with male fetus to demonstrate the point. And so for this particular cancer, we're fortunate that we have this virus called Epstein-Barr virus, or EBV, which is associated with this tumor. And I was wondering whether in the course of this biology, the tumor will release the EBV virus DNA into the bloodstream. And we did that and find that indeed, the patient with this cancer, the blood is really stuffed full of this virus DNA, and healthy individuals are mostly negative. And then I thought that it would be useful to develop an early detection test based on this. It is worth doing because like many cancer, for this cancer, the earlier we detect it, the better is the prognosis. Now for this sort of graph, every time somebody dies, the graph will go down a little bit. Now, for example, if you detect the cancer in stage 1, which is this blue line on top, then by and large, over long term, over 90% people will survive. But if you detect in stage 4, as in the bottom here, then over long term, 40% of people will die. But unfortunately, in many places, including Hong Kong, maybe some 76% of this cancer is discovered late, like in stage 3 and stage 4. And many people are dying. So we want to do something about that. So we decided a few years ago to do a large-scale study in which we studied over 20,000 subjects in Hong Kong and to screen them using this new technology. And we were able to find 34 cases of this cancer. Now, this number 34 is interesting because the incidence of this cancer in Hong Kong is 35 per 100,000. So if you look at 20,000 people, theoretically, you only get 7 cases of cancer. But here, we got 34 cases, which is basically like the cancer, which is coming to be seen over the next 4 or 5 years. So in other words, we predicted that maybe we would be getting some of those cancer earlier than is otherwise possible. Now, indeed it is. For example, without screening, most of the cases in Hong Kong is in stage 3 and stage 4, the orange and red signal. But now, by screening with this technology, most of the cases in stage 1 and stage 2, which is the green and blue signal. And because we're getting them so early, of the 34 cases, only 1 person died. The rest are essentially cured. But however, if you don't use this screening in the bottom here, over long term, some 40% of people will die. So this test is now launched in Hong Kong. It's called the prophecy test. But this nasopharyngeal cancer is only the first step. We're now also working for this technology to be applied to other cancer. But this is more challenging because not all cancer is associated with virus infection. Now, normally you try to detect the DNA of a tumor. One easy way is to use genetics to look at DNA mutations. Like this sequence origin is a T, and now it's mutated to a T to an A. But the problem is that with this sort of mutation detection, it's usually possible if you biopsy a tumor, you know that somebody has a cancer. But in a situation when you don't even know the patient has cancer, this is difficult. So we decided to move from genetics into something called epigenetics. So for epigenetics, basically the DNA sequence doesn't change, but there are some modification of the genome which has changed. It's almost like a room that you're in. I can paint the wall in different colors. I can have different furniture, depends on how I want to use that room for. And similarly in this case, the modification of the same genetic sequence will determine whether a cell might be cancerous. So we have developed this epigenetic test, and we show that it can be applied to multiple types of cancer. And also, excitingly, different organs in our body have different epigenetic profile. So I can also use the same test to tell us where is that cancer. And this technology has been highlighted by MIT Tech Review in 2015 as one of the top 10 breakthroughs of that year. So once again, we decided that this technology might save life, and so we're going to push it out in the clinic. So I co-founded a company called Serena, basically trying to detect cancer at an early stage when we can do something about that. And interestingly, in the Silicon Valley, another group of scientists have also founded a company called Grail. And you can see the mission is very similar to us. So basically, the two companies are moving towards the same direction. So in 2017, the two companies merged together, and I've become a scientific co-founder of Grail. So here is basically at the party celebrating our merger with the first CEO of Grail, called Jeff Huber. So he's a person who actually brought you Google Maps. So after a few years of work, we have now actually launched this test called Gallery test, which can test for 50 different types of cancer in one go. It's a first generation multi-cancer early detection test. So the question now is, what's next? So I think despite what we have done to date, I think we're only seeing the tip of the iceberg. There's still many other genetic signatures in our blood which remain unseen. Now, for example, when we do DNA sequencing of our blood, currently we use a sequencer usually made by a company called Illumina, which is a short-read sequencer. So as a result, people would typically tell you that the DNA in our blood are very short. But I was thinking, would the use of this type of sequencer color or bias what we see? So more recently, we've actually started to use some of the newer generation single molecule sequencer. And for the first time, we can actually see an entire population of super long DNA, walking to tens of kilobase. And so, for example, you try to sequence a fetal genome using this way, the short molecules, you need the hundreds of millions or billions of reads. But now with a super long DNA, I can just use, say, maybe 30 or 40 million reads to do that. So basically, what we've been talking about is for the last 25 years, we've been seeing short text messages from a baby and from cancer sent to us. But now with the discovery of the super long cell-free DNA, for the first time, we can see entire Word documents from a baby or the cancer. So in summary, I think it's worthwhile, if you're interested in research, to think about researching into the non-obvious. Now, for example, like when people talk about DNA being inside cell, I've deliberately looked at DNA swimming outside cells. And also, I think that is useful to have individuals who are clinically qualified and who are also scientists, the so-called clinician scientists, because the clinical side of us will know what the patients need. The scientific side of us will know what is possible technologically. And I also think that it's useful to get inspiration from everyday lives. In my situation, from cooking or from watching movies. And also, it's interesting to see similarity or hidden similarities between apparently different phenomena, like pregnancy and cancer. And finally, I think it's also worthwhile to think about the synergy between academic research and commercialization. Because as scientists, if we're involved in the commercialization, this will allow us to have a chance to influence in what form it will take. And finally, I'd like to thank individuals from my group for generating the data which I present to you today. Thank you very much for your time.